Hello everyone! Welcome to our BMS543. So in this video, we are going to talk about the preservation of microorganisms. So here is our group member, Nur Adila binti Jamaluddin as our group leader, and with the help of the rest of the group member, Umi Nabilah binti Mat Yusof, Nur Hazwani binti Muhammad Husainin, Nurul Naziha binti Zaiful Bahrina, Siti Suraya Maisara binti Abdul Sahar, Nuraida Nabila binti Yusuf, that is me, Nur Azmina Akila binti Syamsul Bahar, Nurul Atika binti Abdullah, Nur Ain Najiha binti Rizwan, Siti Nur Hasina binti Muhammad Iqbal. Today, we will be explaining about the preservation of microorganisms. So here is the introduction for our chapter 3. As you guys know, microorganisms are the valuable biological resource and closely related to biosafety, human health, environmental protection, and renewable energy. The important roles of these microorganisms to preventing and controlling infectious disease. Here we are going to tell you several methods to maintain and preserve the specimen purity and to prevent degradation over time. It's very important to maintain this microorganism. Why? To make sure no contamination and we doesn't need to do it all over again. So here is the 3 objectives of the preservation of microorganisms: to stop the cell division at particular stages. Number 2, to maintain the bacteria culture for extended time in viable condition without any genetic changes. And number 3, to avoid contaminations of microorganisms.
Hi, my name is Nura Atika binti Abdullah. My role is to present about the introduction to the preservation of microorganisms. There are 2 types of preservation. The first one is short-term preservation and the second one is long-term preservation. What is the difference between these 2 terms? Short-term preservation is usually used by university and learning institute because the culture microorganism will only be used for about 1 month to 1 year and the student need to learn on how to culture the microorganism without contamination. Long-term preservation is usually used by biobank organization like Biobank Japan and Genomics England. The method were used by biobank for their research in finding medicine for certain diseases that take years to complete or to study function of certain genes. There are 2 methods that were used in short-term methods. The first one is frequency of transfer and the second one is immersion oil or paraffin method. For the long-term methods, the method that were used are lyophilization and cryopreservation.
My name is Nur Azmina Akila and I will present about short-term preservation method. Frequency of transfer. First, the simplest approach for preserving microorganism short-term viability. Second, microorganism will be safe for more than 1 week. Third, to increase the likelihood of mutation with undesirable changes. Fourth, to support the survival of microorganism and also minimize the metabolic processes. For the second method is immersion oil or paraffin method. Immersion oil method is an alternative method to capping tube by adding a layer of mineral oil to the top of the specimen. A layer of mineral oil at least 1 to 2 cm deep is added. From this method, bacteria and fungi can be stored for a period of up to 3 years. Next, agar slant culture. For this method, which is agar slant culture, prevents the agar and culture from drying out. The cap also prevents airborne contamination from entering the slant. And then, Slanting the surface agar gives the bacteria a greater surface area on which to grow in the test tube. Slant create in the test tube can be kept, which can minimize the water loss. Next, for another short-term preservation method is refrigeration. In refrigeration method, bacteria can survive for a short time for 4 degrees Celsius. In this method can be applied for short duration between 2-3 weeks for bacteria and 3 to 4 months for fungi. At the room temperature, the metabolic activity of microbes slow down and greatly, and only a small quantity of nutrient will be utilized.
Hi, I am Nabila, and today I'm going to share about storage at low temperature, agar slant culture, and liquid nitrogen. In this lesson, you will learn about two methods of storage at low temperature, which is agar slant culture and liquid nitrogen. Let's go first to agar slant culture. What is agar slant? Agar slant is a screw cap culture partly filled with liquid agar mix such as nutrient agar, R2A agar, and tryptic soy agar TSA, and allowed to solidify at a particular angle from 0 degree to 90 degree. The slant increases the medium surface area designed to allow for more growth. Slant means the slope or lean in a particular direction or oblique direction with an fixed angle. And this method is suitable for organisms such as spore formers, yeast, and fungi, and it remains viable for weeks up to months. What is the main function of agar slant? Agar slant is used to culture bacteria cells for identification, and it is used in growing and storing both aerobic and anaerobic microorganisms. Next, let me introduce to you paraffin method, or preservation by overlaying culture with mineral oil using agar slant. This method is using sterile liquid paraffin where it is poured over agar slant culture and followed by store upright in room temperature. Microorganism culture inside the agar slant is maintained by covering agar slant by sterile mineral oil or liquid paraffin. This method is limiting the oxygen access that reduces the microorganism's metabolism and its growth, as well as cell drying during preservation. Next, have a look at the procedures on liquid paraffin method. There are 4 steps involved in this method. Finally, let us review the advantages and disadvantages of using agar slant in preserving microorganisms. The advantages of agar slant is that the slanting surface of an agar provides the microorganism a greater surface area that allows them to grow in a test tube. The slants are created in test tube that can be kept to minimize water loss in order to maintain high moisture content of agar media. However, there are the risk of contamination and the change of physiological and genetically characters due to the growth of mutated microorganisms. Now, let us continue to the liquid nitrogen. What is liquid nitrogen? Liquid nitrogen is a form of the element nitrogen that's cool enough to exist in a liquid state and is used for many cooling and cryogenic applications with negative 196 degrees Celsius. Liquid nitrogen usually exhibits a very low microbial count during production, but during storage and distribution it may become an effective media for the cryopreservation of fungal spores, yeast, bacteria, and viruses. And liquid nitrogen is stored in special insulated containers that are vented to prevent pressure build-up. Depending on the design of the dewar flask, it can be stored for hours or for up to a few weeks. Why using liquid nitrogen? It is because liquid nitrogen is chemically inert,
which means it is not chemically reactive, and liquid nitrogen is also relatively low cost and non-toxic and also non-flammable, and it is readily available. So, what are the functions of liquid nitrogen? Liquid nitrogen can be used for the freezing and transporting of food products. It also can be used for the cryopreservation of biological samples such as sperm cells, eggs, and animal genetic samples. Liquid nitrogen also can be used as a coolant for superconductors, vacuum pumps, and other materials and equipment. And lastly, it can be used in cryotherapy to remove skin abnormalities such as warts. Let us have a look at liquid nitrogen handling. The first one, safety practices. Remember, only store liquid nitrogen in containers with loose fitting lids. It is because a tightly sealed container will build up pressure as the liquid boils and may explode after a short time. Remember, never touch non-isolated vessels containing cryogenic liquids and use tongs to withdraw objects immersed in a cryogenic liquid. The second one is personal protective equipment. Loose-fitting thermal insulated or leather clothes, long-sleeve shirts and trousers without cuffs should be worn while handling liquid nitrogen. A full face shield over safety glasses or chemical splash goggles are recommended during transfer and handling of cryogenic liquids to minimize injuries associated with splash or explosion. And lastly, this is the advantages and disadvantages of using liquid nitrogen in preserving microorganisms.
Hi, Assalamualaikum. My name is Adira and I'm going to present to you about short-term storage of dried culture in dehydrated form. For dried culture storage properties, the bacteria suspension placed in Portuguese and forced air incubator for 24 hours at 25 degrees Celsius. Figure 3.2.1 shows the properties of short-term storage of dried culture. There are two conditions, which is agar plate and step culture. For agar plate and step culture, we must store it at 4 degrees Celsius, while agar plate can store approximately 4 until 6 weeks, while step culture approximately 3 weeks until 1 year. Agar plate streaking is for daily or weekly use. The streaked agar will be wrapped with plastic or sealed with paraffin. The agar then will be stored upside down, which the agar will be on the upside, because it's to minimize contamination and keep culture and agar dehydrated. Figure 3.2.2 shows the method of streaking the agar. Lastly is step culture. Step culture occur on some bacteria strain. The step culture are similar with streaking culture, but the culture is kept in a tube. The step culture prepared by sterilizing strain compatible like lysogyny broth. The liquid warm agar transferred into screw cap vials via proper aseptic method. When the agar solidified, we will pick a single colony from active growing culture using sterile loop. The loop then plugged into soft agar deeply for several times, and then we incubate for 8 to 12 hours at 37 degrees Celsius with the cap is slightly loose and sealed tight. And lastly, we store in the vial dark at 40 degrees Celsius. Thank you.
Hello, my name is Nurhazwani, and you can call me Nani, and I will explain about cryopreservation method.
What is cryopreservation? Cryopreservation is a long-term technique which use of ultra-low temperatures. The cryopreservation method, it is involve liquid nitrogen. The temperature of liquid nitrogen is negative 196 degrees Celsius that can stop all the metabolic activities of the cells, and the sample can then be preserved in such state for extended period. Why cryopreservation? It is because the tissues or cell, which is used for basic research as well for many medical applications, cannot be stored with simple cooling or freezing for a long time because ice crystal formation, osmotic shock, and membrane damage might be occur during freezing and thawing, and it will cause cell death. Thus, the cryopreservation of cells and tissues has been successful method to preserve the tissues or cells with the use of the cryoprotective agents and temperature control equipment. Step of cryopreservation. Firstly, selection of microorganisms. The microorganisms are enucleated into a medium, and the culture are allowed to mature to the stationary phase before being harvested. After centrifuge, the supernatant are discarded and the pellet is withdrawn and resuspended in 2 to 5 ml of broth with the appropriate concentration of cryoprotectant additive. After that, addition of cryoprotectant. The broth that containing the cryoprotectant agent, which is addition of 10% of glycerol and 5% of dimethyl sulfoxide (DMSO), is placed on the surface of the agar. The surface is scrubbed with a pipette or sterile loop to suspend microorganisms, and then the broth mixture is pipetted directly into freezer vials. The function of cryoprotectant is to protect microorganisms from damage during the freezing process, during storage, and during thawing. Cryoprotective agents are often added to the culture suspension. Third is freezing. The freezer vial is kept inside the freeze chamber. The American Type Culture Collection (ATCC) is recommend slow, controlled rate freezing at a rate of 1 degree Celsius per minute until the vials cool to a temperature of at least negative 30 degrees Celsius, followed by more rapid cooling until the final storage temperature is achieved. After freezing, store the vials in the liquid nitrogen. It is recommended that vials be placed initially in a negative 60 degrees Celsius freezer for 1 hour and then transferred into liquid nitrogen. After that, in thawing step, working quickly so that no cell thaw while they are out of the liquid nitrogen storage tank. Place the vials in a cup of dry ice to maintain them cool in transit to the water bath, and then thaw the vial cells in 37 degrees Celsius water bath quickly. Be careful not to submerge the vials as this increases the risk for contamination. After that, placing the vial in the hood. Work quickly. Use a small pipette and pipette the content of the vial to the falcon tube, and then carefully add 10 ml pre-warmed cell culture medium to the tube. Start drop by drop, then increase your rate in order to avoid osmotic shock. After that, post-thawing. This step for cell recovery by serial dilution method to measure the viability of cells.
Assalamualaikum and a very good day. My name is Nurul Naziha and I am going to continue the presentation with freeze drying method. What is actually freeze-drying method? It is a standard multi-step process that is used to stabilize, store, and increase shelf life of biologicals. It is based on lyophilization, which is a low-temperature dehydration process, where it involves freezing the sample, lowering the pressure, and removing ice by sublimation with the help of lyophilization medium. What is lyophilization medium? It is actually a medium that contains lyoprotectants and matrix agent that helps bacteria to survive the freeze-drying process. Lyoprotectants such as sucrose or trehalose will stabilize the cells when water is removed, whereas matrix agents such as mannitol and serum allows the entire sample to retain its shape during and after process. Now let's move on to the steps in freeze-drying method. The first step is sample preparation. In this step, cells are grown whether in liquid or solid cultures form. For liquid cultures, when the cells are in stationary phase, the cells are centrifuged and then the pellet will be suspended with lyophilization medium. Whereas for solid cultures, when the solid cultures are dense, the plate will be flooded with 5 to 10 ml of lyophilization medium. The second step is freezing, where the sample will be freezed down to -40 degrees Celsius, approximately 30 to 60 minutes. The third step is primary drying. This is the longest phase in freeze-drying method. The phase is associated with sublimation, where the pressure is lowered and temperature is increased dependent upon the lyophilization medium used. Primary drying is important to keep the sample colder than condenser but still sufficiently warm so that the water sublime rapidly. The final step is secondary drying, where heat is pumped into the samples for 1 to 2 hours to reduce its moisture levels. It is important for the long-term viability of the cells. After all steps are done, we have to check the cell viability by comparing the samples with original culture. We need to monitor the viability of cells by testing it at 30, 90, 180, and 365 days. A good protocol will yield nearly 100% viable cells. However, if percentage is above 50%, it is considered acceptable.
Assalamualaikum and hello, I am Surya and today I am going to talk about spray drying method for preservation of microorganisms. As we know, we have two types of drying methods in preservation of microorganisms. One is short-term and another one is long-term. And this is the long-term part of the drying method. So it is the cost-effective technique that popular in storing probiotics and drugs in pharmaceutical industries. So moving on, what is spray drying method? Let's find out. Spray drying method is the preservation of microbial technique that is continuous made and can be carried out in industrial scale. I'm so excited to tell you this because Yakult using this method to obtain the good bacteria in their drink. And now the parameters. Viscosity, high viscosity for correct drop formation. Solid content and nozzle material. Solid content must be above 30% and nozzle material must have excellent resistant abrasion and good corrosion. And then surface tension. Small amount of surfactant, lower surface tension. Feed temperature is required high. Also volatility solvent is required high. And then this is the illustration of spray drying machine. Look at those components. Moving on to the real life one. So impressive and big. Wow, right? But what about the method? Let's learn about the methods of spray drying. Okay. At the initial stage, the liquid is dried atomized into good droplets. It being placed regularly, relocating stream of warm gas. The temperature levels of the droplet is gradually increased and also good droplets obtained dry easily. The droplets are easily vaporized and turns into solid fragments. First, move to the separator with the drying fragments. This is where it gathered. The cyclone separator is to make sure the very good airflow and facilitate heating and mass transfer, and make sure that the dry fragments are divided by centrifugal activity. So that is for spray drying method. Thank you. I hope you guys enjoyed this and learn more about spray drying method. Thank you. Bye-bye.
So this is the reference that we refer for our video. Thank you so much. Hope to see you again, and not to forget, credit for our beloved Madam Mazatul Ikma Mak Zain. Bye bye. See you guys soon.